Hey, James, we need about 1 minute, Labrina. A little bit late, but we're coming. All right.
What?
I've been wondering what the fridge keeps smelling. I've emptied it 4 times.
I see. Claire did try and get Lewis to trade. No.
No, it's definitely in it. It's only when you open it. Okie doke. Are y'all here? Because I'm starting.
Lewis's reaction when he.
Yes, ma'am.
Oh,
Oh, you're right. You found a brownie point. Why even started?
like he almost.
Look at that.
Isn't today lesson 10?
Bailey just caught it. Yeah, it is. And I just didn't. That's a good one.
Curled.
There was no homework.
It was doubled over.
On my homework page is just blank.
It was,
James entered the lane.
Something was due on 26th, that was all.
Claire bucketed
it's
Jared
so it's
water.
that, crossword you already did.
That was last week.
It was
The
just,
salt was not that bad.
it was,
That was lesson 9. Lesson 10 is just blank.
because less than 9, you bring them home. Yeah, it's,
Mary didn't even want to do it.
hang on.
It's not due till next week.
I think
But
It
I gargle with
is,
salt water
it's
if
in
I have,
teacheries.
like, a sore throat or something.
It's-
So I
yeah.
guess I'm just
Okay.
Yep. All right, so let's start. So y'all bear with me. Because like I said, I still have some laryngitis from the cold I had over the weekend. And I really am, I'm trying not to yell or push my voice too hard, which I've already wrecked today. Already failed. I've already failed at that, but we're gonna try. All right, so there's James. Hey, James. Sorry, we're a minute late, buddy. All right, so lesson 10, DNA. So we're talking about identifying the substance of genes. So there was a time when we knew there was some substance that's getting passed from parent to offspring, and it's carrying information. It's carrying information about how to build that offspring, but we didn't know what it was. And then once we knew what it was, we didn't know how it looked. So this kind of goes through the whole process of all the steps for discovery. So the first thing we worked with was bacterial transformation. So this is the middle of the 1900s, and this is when scientists started asking questions about genes. So we're talking, you know, really just a little over 100 years ago is when this started, this whole quest to figure out what's going on with genes and DNA. So you might know somebody who was alive in this time period. Yep, your great grandmother. Yep. Mhm. So it's not, this is relatively new stuff compared to a lot of things in science. This is pretty new. So we wanted to know, what is a gene made of? How do genes work? And how do genes determine the characteristics of organisms? These are the things we need to find out.
all I know is Levi's makes them the best.
Levi's makes them the best.
Yes,
There you go.
that and Wrangler.
So here's what we knew at the beginning. We knew that a section of DNA on a chromosome in a nucleus, that there is a section of DNA on a chromosome in a nucleus. That much we knew. And that each chromosome contains hundreds of genes. And that contains those contain most of your characteristics, your hair color, skin color, how things taste to you, all that stuff. And they contain instructions for making specific proteins.
Not going to.
And that is by Ozzy. Thank you. Ozzy,
People have food.
come here. And that's determined by a sequence of amino acids and the order in, I know, I'm sorry. And the order in which those amino acids are strung together to build a protein. So we started with bacterial transformation and this dude, Frederick Griffith. So this is 1928. So not that long ago, really. So what he did is he had 4 different kinds of bacteria and he injected those into these lab mice, okay? So he had heat-killed disease-causing bacteria. that he injected into mice. So bacteria that he knows causes disease, specifically pneumonia. He killed the bacteria with heat and he injected that into the mice. And then he had a live bacteria that was a harmless one. It didn't cause the disease. And he injected that into the mice. And then he had 2 strains that together caused a fatal pneumonia. Ozzy, no. So he concluded that the genetic information could be transferred from one bacterial strain to another. And he calls this transformation. And it's when one type of bacteria, which is harmless, is changed permanently into another, which was disease causing. The transformation factor must be a gene, he says. Yes.
Diseasing causing.
Where?
I was like, is that a word?
Diseasing
Diseasing
causing?
Diseasing causing.
Oh,
Yeah.
yep. Hold on.
Yeah,
Good one.
Jack found it. I'm like, I'm not smart, but
Supposed to be a hyphen in
he
there.
pointed it out to me.
Cool. Good one. All right, so then this is how his experiment looked if you were to make a nice little infographic out of it. So this live strain, R strain, mouse is living. S strain, a mouse dies. Heat killed strain, S strain. Awesome, he lives, even though otherwise it would have killed him. And then when you put them all together, the mouse died. So he's saying that a live S strain from group 4 made it into these mouse that died. And he was able to determine that by blood analysis. And this is our buddy, Frederick Griffith, okay? Very stern looking fellow, very stern. Reminds me of the principal from Back to the Future. Doesn't he look like him? Yeah. What? Yeah, exactly right. Yes. You're a slacker. Anyway. So then the next thing was to determine if it was only the DNA that causes this transformation. So this is where Oswald Avery came in. So Oswald Avery, he discovered that DNA stores and transmits genetic information from one generation to the other, and that DNA is what's responsible for this transformation that Griffiths had noticed. And this is Avery working with Colin MacLeod and Macklin McCarty. So these are some serious Scottish people here. And this was in 1944. So if you have grandparents the age of my parents, they may have been born in a time when we didn't even have a clue about DNA. So if you think about just in a lifetime, how we can go from very rudimentary methods like electrophoresis and identify a person when we didn't even know what the stuff was, 70, 75 years ago. So like my dad's born in 1942, my mom in 1946. So that's how recent this is. So what they did basically was a process of elimination. They wanted to know whether it was a protein, a lipid, a polysaccharide, an RNA, or a DNA that was causing these changes. So in order to do that, they basically removed everything one after another, this process of elimination, until they determined which one of these substances was responsible for the transformation that Griffith had seen. And by process of elimination, it was DNA. So now we know it's DNA, but we don't know what DNA looks like. We don't know what's in it. I mean, we know some things. They know that they've named it deoxyribonucleic acid, so they know what components are in it, but they have no idea really what the structure is. So these are those dudes. This is Colin MacLeod, Macklin McCarty, and Oswald Avery. They look a little more fun.
The two on the right look like they're having a good old time, talking about the old days, and the other guy's like, I wonder how much DNA is in that bird.
He looks like a meteor is coming,
He's like, hey guys,
They're
that
the ones who
meteor.
named it Geo.
Well, I don't know, did they name it? I think, no, I don't remember if they named it or not specifically. Good question. Can't remember. So now we have bacterial viruses. Okay,
It won't stay.
these are bacteriophages and they're so cool. So these things are really fun. So a virus that can infect a bacteria is called a bacteriophage. And they actually do look like that. They look like some kind of weird lunar lander alien spaceship. They're cool.
I want to be that one.
right? So basically what it is it's some DNA surrounded by a protein envelope. And yes,
War of the Worlds.
oh yeah, yeah. So see how it has a little landing legs and this little pointy thing here? So it will come and land on a bacteria cell, land on a bacterium, and jam this thing, pop, inject it right into the bacteria cell and inject its own DNA, which will force the bacterium to replicate its DNA, which is what they do, right? So these are viruses, but they infect only bacteria.
It's like a spider in a spaceship.
Mhm.
Baby.
So all bacteriophages are composed of a nucleic acid molecule that's surrounded by a protein structure.
And keep it a syringe.
The bacteriophage attaches itself to a susceptible bacterium and infects the host cell. Following the infection, the bacteriophage hijacks the bacterium's cellular machinery to prevent it from producing bacterial components and instead forces it to reproduce viral components. So eventually, new bacteriophages assemble and burst out of the bacterium. That's called lysis when it breaks open. Here comes a whole army of them. And bacteriophages occasionally remove a portion of their host cell's bacterial DNA during the infection process, and then transfer this DNA into the genome of a new host cell. And that process is called transduction. But these are used in research a lot because they're very simple. Whoop. So this is where Hershey Chase came in. So, whoop. So Hershey and Chase. So they studied a bacteriophage composed of a DNA and a protein coat, right? And they wanted to determine what part is entering the bacterial cell. So they know that bacteriophages are viruses that infect bacteria, but they don't know whether it's the protein component or the DNA component that is getting into the bacterial cell and doing the infecting. So what they did was they used radioactive markers and they labeled the DNA and the protein. And then they were able to follow it on its journey into the bacterium and determine what was going in versus what was staying out. So here's a graphic for that one. So this they used in this case radioactive sulfur 35 and radioactive phosphorus 32. And they blended them up. Look, there's a blender, right? And they centrifuged it. And in the end, after following the radioactive components, they were able to determine that it's the nucleic acids that hold information for heredity, because the bacteriophages were passing their genetic information on to the bacterium. And the only way that's happening is if DNA is what contains the genetic information. So this is Martha Chase and Alfred Hershey. They're hanging, being cool, right? So then the next thing we're trying to figure out is what role does this play for sure? Like really, what does it do? So we know that it makes up genes, and if it makes up genes, it's got to be capable of some things. It's got to be capable of storing information. It's got to be capable of copying. It's got to be capable of transmitting, and the genetic.
That's wrong. That's a brownie
I think it should say all, but I caught it, not you.
But there's also a comma there.
Yeah, I copied this from something, but I'm not sure what it was supposed to be part of. So, anywho, transmitting all the genetic information in the cell. So this is what it has to be able to do, right? So we don't know how it does it yet. Okay, so what happened when Griffith injected mice with pneumonia-causing strain of bacteria that had been heat killed? That's right. They stayed healthy. And what happened?
When you say we don't know it yet, do you mean
In this,
or at that?
no, right here, where we are in this lesson. We don't know yet.
Okay.
We do know now.
Okay.
All right, what happened when he injected them with a mixture of heat killed and pneumonia causing bacteria and live bacteria?
They died.
They got pneumonia and they died. So what was the purpose of Oswald Avery's experiment?
To prove the other guys, right? They were trying to prove him.
Yeah, basically they're backing up Griffith. So they wanted to find out which molecule in the heat killed bacteria was the most important for transformation. They're following up on Griffith. Something's going on. They want to know what it is. So what did Avery do? He used enzymes, destroyed various molecules in the heat-killed bacteria, including lipids, proteins, carbs, RNA. And transformation still occurred until he used enzymes that broke down DNA. So he used process of elimination to figure out what was what, right? And what did he conclude?
DNA stores and transmits.
That's it. Yep. Okay, so Hershey and Chase, what did they conclude?
That DNA is what stores and transmits it.
Right, so they worked on the bacteriophage. So they said it must be the genetic material for sure, because it's what the phage is injecting, not the protein. Yeah. So how did they confirm Avery's results? They basically said that DNA transmits genetic material from one generation to the next. Hershey and Chase concluded the genetic material in the bacteriophage was DNA, not protein, confirming Avery's assertion. So if you look here, what's this thing called?
Bacteria.
It's a bacteriophage, right? All right. So if this one has DNA with the radioactive label,
Yeah, that's the phage.
and what's happening in this step?
Injection.
Right.
Injecting.
It's going to infect this bacteria. And what's it going to do here?
Run away.
Run away? No. So it's if it has a radioactive label on that DNA, now you have radioactivity inside the bacteria, right? So in this case, see right there, there's the radioactive label on the DNA. Now the radioactive label is on the outside, so it's on the protein. So when it injects the DNA,
Run away.
the radioactivity, does the radioactivity go into the bacterium? No.
Run away.
Leaves with the bacteriophage. So here's this chart, if you had it. So for storing information, it's an important function because DNA makes up the genes that control the development and characteristics of different kinds of organisms. That should have an S. I see it. Instructions to, an example would be like instructions to cause a cell to become a giant oak tree in time. Starts out as one cell, right? All right, so why is copying information important? It's got to be accurately copied every time a cell divides. And after mitosis, each new oak tree, for example, has the same genetic information as the parent cell. That's important. You don't want something completely different coming up. And then for transmitting, why is this important? So genetic information must pass from one generation to the next. Whoops.
Drop the pencil. It's under the table.
Bye, Anthony. And gametes of the oak tree carry genetic information from the parent to the offspring. So the offspring develops into the oak tree. So now we get into the fun of trying to discover the structure of DNA. So what they knew at the time was the DNA had certain components. They knew it had a 5-carbon sugar, which is deoxyribose. They knew it had a phosphate group. And they knew it had 4 nitrogen bases. And they knew that these were in 2 groups, purines, which are adenine and guanine, and then pyrimidines, which are thymine and cytosine. A goes with T, G goes with C. Purine with pyrimidine, purine with pyrimidine, vice versa, right? Okay, so we know all these things are there, but at this point, we don't know how they fit together. So then we get this dude, Edwin Shargraf. So this is 1949. So this is even more recent. We're getting even more recent. So he found something that's really key. With his research, he was able to determine that there's always equality in the quantity between A and T and G and C. So why is that so important to know, Alex? Why is that so important to know? That there's always equality in the quantity of A and T and G and C.
It helps narrow down how many possible things there are.
Mhm. So if you know that the quantity of A and T are always the same, and the quantities of G and C are always the same, it tells you something about the relationship between A and T.
You infer that they're always together.
They're always together, yeah. If you have one, you have the other.
Oh, I thought it was there was an equal number of A's and G's.
No, A and T,
Okay.
And
not
G and
A,
C.
T, and G.
right. So by discovering that the quantities are always the same, you know that if you have A, you're going to have 1 T for every A, basically. And then you're going to have 1G for every C and vice versa. You've got a ratio thing going on. And that's super important to understand when you're trying to come up with a structure. Okay. So next we get this dude, the same dude. This is Edwin Shrugroff. He's the one who came up with that.
He looks really.
He looks friendly to me, like the kind of guy who would give you a Werther's
like-
caramel, you
He looks
know?
like an old time radio.
Yeah.
host.
Yeah. He looks pretty cool.
And Jay Mann.
So now we get to famous, yes, photo 51, which I'm wearing. So one of my favorite science photos ever. Rosalind Franklin. Now pay attention to the date. 1952. This is important. So photo 51 was taken in her lab by her lab assistant, who was a postdoctoral student named Raymond Gosling. Okay. Yeah. So he's a grad student. He's working in her lab, and she has set up all this apparatus to do X-ray diffraction pictures. That's what this is. And it took hundreds of hours to get the apparatus set up, to run all of the, preliminary piloting to get these pictures. Now, this picture is of DNA. This is an actual picture of DNA, but it is from the top down, looking through the helix, like looking through a tube. It's not from the side, which is usually how we see DNA illustrated, right? So it's looking down. So the outside of the chain has a backbone of alternating deoxyribose and phosphate moieties. And then you got base pairs that code for protein building, and that they're inside of the helix. Now, this is important because you see this X here, that's where the helix crosses. And then if you look closely, you can almost see the beginning very faintly. See, like right here and right here, that would be the next layer of the helix. So you've got a ladder and you twist it and you look down through the middle. So you're going to see a lot of these little all the way down, right? This is super important because nobody had a clue about this double helix structure. They've been using models and trying to make it work, and nobody could make it work. So this is Rosalind. All right, and if you remember, I've told you about Rosalind before, but Rosalind was not a warm, fuzzy person. She was kind of abrasive.
I think she was deceiving.
And well, she was kind of abrasive. She was brash. She was not well liked by her colleagues. And part of that did have to do with her being female in a time when labs were predominantly male. And it was definitely a boys' club. And she didn't meet the standard for how a lady was supposed to act. She didn't dress the right way. She didn't talk the right way. She didn't behave the right way. She wasn't genteel at all in the 1950s when women were expected to be. She was just kind of a tell it like it is person. And she was not well liked. So she didn't get a lot of support from people. So then we have this dude, Maurice Wilkins. And I've given you a little spy for him because I want you to remember this. So now check her date. It's 1953. Now, granted, Rosalind has taken this picture and she knows now that this picture, she knows now what she's seeing, but she's going to take the time to do the math necessary to come up with all the proper calculations to spell out exactly what that layout looks like, because she's a very specific person. So she knows now. In 1952, she knows the structure of DNA, but she's not going to release it until she has all the math to back it up. Okay. Now this dude, remember this dude. So the next we get Watson and Crick, 1953. Watson and Crick were using a model. This is the actual model. There's little sticks and balls and things like that. And they had organized that to look like this one looks like the shape of adenine, and this is the shape of deoxyribose, and this is the phosphate. But they could not figure out how to put it together in a way that made sense. They've been playing with the model for, it's like building Legos for a long time. So what they did was they used the evidence from Rosalind Franklin's picture, photo 51, this picture, to determine the structure of DNA. And where'd they get it from? You remember our buddy Maurice?
Was he
Yes.
an imposter among us? Whoa.
So this is Watson and Crick standing with their model, looking very proud of their double helix, which you can very clearly see. It's pretty cool, right? That's pretty cool. But Franklin as you'll learn when we watch this really quick little TED-Ed, Franklin did not get the acknowledgement for this. And it's really only been maybe the last 15 years that she's gotten proper credit for what she did. So I know in the 90s, when I was studying biology, she was mentioned, but the credit wasn't given to her at the level it should have been. So we're talking from 1953 till probably close to 2000, 2010, before she really got proper credit. Yeah, Maurice was not a good guy.
What a dream.
Yep.
Discovery of the structure of DNA was one of the most important scientific achievements in the last century in human history. The now famous double helix is almost synonymous with Watson and Crick, two of the scientists who won the Nobel Prize for figuring it out. But there's another name you may know too, Rosalind Franklin. You may have heard that her data supported Watson and Crick's brilliant idea, or that she was a plain-dressing, belligerent scientist, which is how Watson actually described her in The Double Helix. But thanks to Franklin's biographers, who investigated her life and interviewed many people close to her, we now know that that account is far from true, and her scientific contributions have been vastly underplayed. Let's hear the real story. Rosalind Elsie Franklin was born in London in 1920. She wanted to be a scientist ever since she was a teenager, which wasn't a common or easy career path for girls at that time. But she excelled at science anyway. She won a scholarship to Cambridge to study chemistry, where she earned her PhD. And she later conducted research on the structure of coal that led to better gas masks for the British during World War II. In 1951, she joined King's College to use X-ray techniques to study the structure of DNA, then one of the hottest topics in science. Franklin upgraded the X-ray lab and got to work, shining high-energy X-rays on tiny wet crystals of DNA. But the academic culture at the time wasn't very friendly to women, and Franklin was isolated from her colleagues. She clashed with Maurice Wilkins, a lab mate who assumed Franklin had been hired as his assistant. But Franklin kept working, and in 1952, she obtained Photo 51, the most famous X-ray image of DNA. Just getting the image took 100 hours. The calculations necessary to analyze it would take a year. Meanwhile, the American biologist James Watson and the British physicist Francis Crick were also working on finding DNA structure. Without Franklin's knowledge, Wilkins took Photo 51 and showed it to Watson and Crick. Instead of calculating the exact position of every atom, they did a quick analysis of Franklin's data and used that to build a few potential structures. Eventually, they arrived at the right one. DNA is made of two helicoidal strands, one opposite the other, with bases at the center like rungs of a ladder. Watson and Crick published their model in April 1953. Meanwhile, Franklin had finished her calculations, come to the same conclusion, and submitted her own manuscript. The journal published the manuscripts together, but put Franklin's last, making it look like her experiments just confirmed Watson and Crick's breakthrough, instead of inspiring it. But Franklin had already stopped working on DNA and died of cancer in 1958, never knowing that Watson and Crick had seen her photographs. Watson, Crick, and Wilkins won the Nobel Prize in 1962 for their work on DNA. It's often said that Franklin would have been recognized by a Nobel Prize if only they could be awarded posthumously. And in fact, it's possible she could have won twice. Her work on the structure of viruses led to a Nobel for a colleague in 1982.
She got robbed.
It's time to tell the story of a brave woman who fought sexism in science and whose work revolutionized medicine, biology, and agriculture.
Right.
It's time to honor Rosalind Elsie Franklin, the unsung mother of the double helix.
I guess it was like a mixture of her not being like
Yep.
going against the grain,
Yep.
and she was a little brash. And
A lot of things. Yeah.
culminated.
A lot of people didn't take her seriously because of her gender. A lot of people just didn't like her because she wasn't nice. And she certainly didn't fit the model. I mean, maybe had she been a woman in a science lab who was also friendly and warm. Maybe she would have had a better chance, but she wasn't. She was just who she was. And Maurice Wilkins completely, yep, absolutely, completely, literally stole her research. And while she took the time to do all those calculations and be specific, Watson and Crick slapped several things
We're like,
together.
hey, why not?
Oh, let's beat her to it. And yeah, and they knew very well what they did. It wasn't as if they were unaware. They were very much aware of what they
She
were
got
doing.
robbed twice when she don't know these guys.
Well, yeah, well, so they're not awarded posthumously. So how did she die? Cancer. Now, what might you infer based on her?
X-ray lab, Cancer.
Correct.
X-ray lab.
Yes.
Cancer.
She actually died of ovarian cancer because she had most likely because she had spent so much time around high energy radiation.
I mean, it took 100 hours just to get that.
Yep.
Yeah, I think she should have worn a lead coat.
Yeah, but they didn't realize then, necessarily that we're
Then they're
talking
like,
about
"Oh, all
damaging
these people are dying.
damaging
Ooh,
your
that's
cells,
not
right?
great.
So the double helix model. All right, so now we know that it's a double helix. So this is kind of like what Watson and Crick had. They had these little pieces like this, right? And then they're trying to figure out how to fit them together. And you can imagine if you have no direction for this, this is really hard. Like this is super. How would you? They could have spent 2 years, 3 years slapping stuff together, never getting anywhere. And they saw that picture and went light bulb, and they knew what to do. So DNA is double stranded. It's a double helix. It has side ladders. That are these pieces here. It has, and the side ladders are composed of your sugar, which is the deoxyribose and the phosphate group. And remember that phosphate group is negatively charged, which is why electrophoresis works. And they're held together by hydrogen bonds. And then it has steps in the ladder, and the steps in the ladder are purines and pyrimidines, which we knew these were the components before. We just didn't know how they went together. Now we do. So the building blocks of DNA are, what do we have?
Nucleic acids.
There you go, nucleotides or nucleic acids. Yep. The nucleotides in DNA are made of 3 basic components. A sugar called deoxyribose, a what? A phosphate group, and nitrogenous bases, right? So DNA contains 4 types of nitrogenous bases, and these are,
Adenine, guanine, tyrosine, thymine.
right, ooh, where's that one? Oh, I don't know why it didn't click.
Wait, what happened to those first two?
They should be there.
That's not a
I think I may have deleted them.
That's not a brownie point.
Yes,
That's
I was
them.
just like,
They're
where
there
are
for
they?
spirit, okay? They're
Yep.
folks. I can imagine.
All right, so, sorry.
You need an X-ray to see them.
In DNA, nucleotides can be joined in any order. And the nucleotides in DNA are joined by covalent bonds, which is good because they come apart nicely, right, when DNA unzips. So what did Chargaff contribute? Remember I said his was really important?
He figured out who they paired up.
Yeah, he knew that they were in equal percentages or equal ratios, A and T and G and C. So for every A, you have a T, for every T, you have an A, and so on. And that's super helpful. Franklin, she had her diffraction photo.
She can, if you did. 50,
Right.
photo 51.
Yeah, and that actually shows your double helix. There are a couple of science pictures that are really iconic.
What'd you
Photo
take the picture
51,
with?
huh?
What'd you take the picture with?
X-ray diffraction. It's X-ray crystallography. It's a whole, you could probably spend a year learning on that. It's,
Probably more.
yeah, clearly. So that one, the one, what's it called? Does anybody know the picture of Earth from one of the Apollo missions? The first one, you can see the whole planet for the first time ever.
Yeah.
That's another iconic one.
The one they just took of the black hole.
Yeah, the black hole. Mhm. But this is funny because it's probably the smallest thing, but super iconic. So, and Watson and Crick, they contributed the actual
Smallest thing that
model, right? They built the
They
model.
contributed- Yeah, that, the smallest
So,
picture.
this
was
is-
the biggest.
They made the model. They made an actual model, and it was big. It was like, it was big. It was big.
So that's how she
Huge.
could fit in the DNA. thing.
Yeah, there you go. No, their actual model was big, a little bigger than human sized. They used it to
Tiny little person.
build.
Every second.
So using Chargaff's rules, which is going to tell you, so here's the thing to know. If you know that your percent DNA sample is 31.5% adenine, then what's the thymine percentage? 31.5. Now, if you know that, you now know the percentage of guanine and cytosine, which is what?
18.5 for
18.5 for both. Exactly. Very good. So how about if we know thymine at 30, then we're going to know that adenine is also 30. And I think, oh, that 20 was, okay. And then cytosine is 20. So how about if you only know cytosine is 17?
And the other one's 17.
Then guanine is also 17 and the others are 33 and 33. Right, they have to be. So looking here at this diagram of DNA, what is number 8 pointing to?
hydrogen bond.
There we go. Yep, I can see it now. Come on, click. Oh,
It was like,
oh,
they just said that it was a covalent
it
bond.
doesn't like it when I click it. So it's the hydrogen bonds because it's pointing to the hydrogen, it's pointing to the ladder.
Hydrogen covalent.
It's hard to see.
Wait,
this. Well,
It
it's,
says nucleotides DNA are joined by covalent
oh, it is
bonds.
pointing to,
Up.
so wait, this
On
one
number
then, so
5.
9, it looks like these are, yeah, I think my book had that wrong because it does look to me like that's pointing to the covalent bond between the bases, not pointing to the ladder.
Oh, so it's supposed to be
The one is.
So if you perceive this picture as pointing to the bond between the two nucleotide bases, then it's going to be covalent bond. But if it looks to you that it's pointing to the ladder, then it's going to be hydrogen bond. So this is the nucleotide. This is the deoxyribose sugar that it's showing you here in the phosphate group,
My thing is,
and then your base pair.
it says that the nucleate in answer in question 5, it says the nucleotides in DNA are joined by covalent bonds.
They are correct.
And then it's also hydrogen bonds.
Are you looking here?
Yeah, and then over question 8 is hydrogen.
Well, that's what I was just saying. I think they intended for 8 to be pointing to the ladder itself, which would be showing the bonds along the ladder, not the bonds between the bases. If it appears to be the bonds between the bases, which is what I see, then yes, it would be covalent bonds. That would be correct. Does that make sense?
Mhm.
This is one of those ones where I look at the book and go, is that really pointing to what I think it's pointing to?
the deoxyribose and the phosphate are joined together with covalent bonds, and then the two nitrogen bases are together third by other way around.
Yes, the other way around. The nitrogenous bases are joined by covalent bonds. The deoxyribose phosphate along the ladder, those are hydrogen bonds, which means they're not going to break apart, right? Because those are stronger. All right, so replication. How does this work? All right, so how do we copy this code? So DNA must get copied before a cell can divide. So this occurs during late interphase, and the DNA is going to unzip into 2 strands. We were just talking about how these bonds right in the middle here are covalent. So they break apart easy. Do you remember when, for those of you that everybody did chemistry, except you. So we had sugar and salt and we heated them. The ionic bonds in the salt, it didn't break apart with just heat, but the covalent bonds in the sugar did break apart. You remember the sugar melted and the salt didn't?
I think that's one.
There you go. All right, so it's going to unzip into 2 strands. And the 2 complementary strands, remember complementary doesn't mean they match. It means they go together, just like the homologous pairs when we talk about chromosomes, like your shoes, right and left. So each new copy has 1 original strand and 1 new strand. DNA polymerase, this is the enzyme that does it all. This is the cool enzyme. So it joins your individual nucleotides to produce a new strand of DNA. So this is a really, really, really good illustration of how it functions. Oh, hush. Come here.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions, This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments.
Named after someone?
Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So you have, this is this process of transcription. All right. So there's going to be transcription and translation, right? Piper, you good? Yes, you're sleepy today. Sit yourself up tall. That'll probably help. Or go sit by Bailey at one of those hard chairs. So replication in living cells. So how do we replicate in living cells? So in prokaryotic cells, there's only one point of origin. Replication occurs in two opposing directions, just like you saw, right? At the same time, and it takes place in the cell's cytoplasm because prokaryotic cells don't have a membrane-bound nucleus, right? In eukaryotic cells, on the other hand, there are multiple points of origin, and it uses unidirectional replication, the nucleus of the cell. All right, so why are the strands of a DNA molecule said to be complementary? Why are they said to be complementary?
Because they have the same amount of A and T as A and C, and they go together.
Right, they go together, they join together. All right, so what's the first step in eukaryotic DNA replication?
Unzipping.
Gotta unzip. Can't unzip, can't do anything.
It's
So-
like zippers that go-
If the base sequences on a separated DNA strand is CGTAGG, what will the base sequence on this complementary strand be?
GCA is that GG?
Yep,
CC.
yep,
So it should read backwards because it's going to upset.
so it's 5' and 3'.
Yeah.
So it reads from 5' to 3'.
Yeah.
So one part of it reads forward and one part of it reads backward. And you have to kind of know, so like when we were looking at the lab, and it was talking about like ATGC or whatever it was, if you noticed on the bottom strand, it was going from right to left, but on the top strand, that same combination was going left to right. So what enzyme joins individual nucleotides to produce a new strand of DNA?
DNA polymerase.
Polymerase. Yep, that's the big one. So if you had, this is that, if you wanted to look at that. All right, so before we do, we could do more dragons, but instead of doing more dragons, which were super fun, I have something else for you guys to do. So we're probably going to probably want a table, but I'm going to show you. So what I have here are little nucleotide bases attached to the ladder. Okay, so for example, this right here. Okay, this is going to be your 5-sided sugar and your phosphate group. I'll hold it up. 5-sided sugar and phosphate group. And here's your base. So this is G. So I've got G and I've got T, A, and C. And the way these work is they fit together. They're going to lock together. And if everybody makes 1 or 2 of these that lock together, then we can lock them all together and make us a big double helix, because the way they fit is going to be double helix. So I'll just show you real quick because it's kind of tricky. And then I'll let them go because there's nothing really for them to watch, but I'll send them pictures. So there's little cuts in these, and it's like this one goes into here, and at the same time this one goes into here. So it is on cardstock, okay, but you still have to be super careful because we don't want to rip them, right? If we can do it without taping them, I would love it because then we can save it. But if we have to tape them, no big deal. So when you get this set together, it's gonna go just like, whoop, one more. Get in there, come on. There we go. So here's how, for example, these 2 are gonna fit, just like this. All right, but then what you have to do is you have to fit the next one. So if this next one is gonna be an A on this side, then I have to bond my phosphate to the sugar, right? So the phosphate's gonna go to the sugar, right? And then on the other side, it's the same thing, but it's gonna go kind of this direction because you kind of have to build that way. And then we'll try connecting them. So why don't you guys here come up and I'll give you each a couple. And then I'm gonna let Shannon and James go, but I'll send you pictures of the final product. Okie doke. Bye. I'm gonna let them go. Hang on. There's really no point in them watching you fight with paper. It's gonna, okay.